All right, good morning, everybody. We are ready for some genetics today. We're going to be doing some a lab looking at monohybrid and dihybrid crosses, so you'll get a little bit more practice with those. And yeah, so I know that there are still some power outages and Wi-Fi outages, and frankly, I was up all night. I've got a grant due today at 5 p.m. So I've been up staring at my screen way too long. So that's why I look like death this morning. So I apologize for all of these technical issues and the fact that we can't just do this in person because it's such a simple lab. But the good thing is that instead of looking at me talking about this morning, you can actually watch an older video that I made in a previous semester to sort of describe what's going on. Now, I know that Blackboard has a really bad sort of relationship with YouTube. And so normally I would try to have everyone kind of watch together. I don't know if that's going to work. I'm going to try it. And then if it doesn't actually work, then we'll just have everyone watch the videos and then do the lab. So the lab itself is, and it is an experiment, but because of the way that we're doing this online, I can't actually like give you an ear of corn. So what I've done in the video is I've counted the corn for you, and so everyone will have kind of the same data. And so when you go into the data tables in the lab itself, all of the answers should be the same because the data is the same. Now, when we do this, I want to make sure that you guys understand why we're doing this. And that is because when you're talking about transmission genetics, you're generally talking about starting with a phenotype. And from the phenotype, you can make predictions about genotypes. And that's really what we're doing when we do these genetic crosses. And so we start with parents that we're pretty sure of what their true breeding genotype is, which is really a true breeding phenotype because you're monitoring the phenotype, you're not monitoring the genotype. And so as those parents pass down these traits or these phenotypes to their offspring, we can kind of retroactively make predictions. That's called, what is that, posteriori data, whatever it is. So so that's what we're doing. And in any field or in any type of transmission genetics, whether we're talking about bacteria, whether we're talking about humans, whatever organism we're talking about, we're starting with the phenotype because that's what you record, that's what you notice, and then we're going to kind of work backwards to see if our prediction is true. So in this case, we are trying to see if, in fact, these are Mendelian traits. Mendelian traits meaning that they have predictable patterns of inheritance of the F1 generation should be hybrid, and the F2 generation should have that phenotypic ratio of 3:1. So when you're filling out these tables with the coins that we used, because the probability was 1 out of 2, that was a little bit easier to work with because it was just a 1/2 probability. But when you get to the monohybrid cross, where you're crossing the F2 crossing to get the F2 generation, because we're counting the F2 generation, you have to consider that we're thinking of the phenotypic ratio, which is not the same as the genotypic ratio, and it's not the same as using coins, because coins don't have phenotypes, unless we said that heads was dominant. right? So because heads and tails are equal in the phenotype of coins, it was 1/2. But the phenotypes are not equal in the F2 generation, so they're 3-to-1. So you would have 3/4 dominant and 1/4 recessive. That's the biggest mistake that most students make when they're doing these calculations. So just make sure that you remember what your hypothesis is, and it has to do with the phenotype. All right. Any questions so far? I know it seemed like I'm rambling. That's just because my brain is somewhere else on the grant proposal. Sorry about that. All right, so let's see. I'm gonna try to share the video and see if it works. And just as soon as I hit play, let me know if you can hear it, because I know that sometimes you can't actually hear it. And I don't know what setting to use to change that, because it's set up for, obviously, for me talking. All right, so let me see. No. All right, so monohybrid cross. Oh, here, share audio. Got it. Okay, so I'm going to start the video. You should have control of the video, but if you control it, then everybody else, then it stops for everybody else. So just an FYI out there that it's not that everybody's seeing the same thing. All right, let me share that. And please excuse the yeah. So today's lab has to do with corn. And you're thinking, corn, hooray, corn is kind of a boring thing. But corn can actually be kind of interesting. For example, corn has multiple phenotypes. And if you can see, we've got this beautiful kind of red and dark red corn here. And then we've got this really cool multi-colored corn cob with some lots of different phenotypes actually in here. We've got your typical yellow and white, but we also have some blue and pearly, etc. And then in this one, we have a nice modifier gene that's causing all of the kernels that would look much brighter color to make them a little dull. And so there are lots of genes that are involved in corn phenotypes. For example, some of the phenotypes are just really small cobs. This is a blue type of popcorn that I grew in my backyard a while back. And I actually grew some more corn this year that has more of these phenotypes in it. But I planted it a little bit too late and it hasn't actually made corn yet. So I have a couple corn babies that hopefully before the end of the semester I'll be able to show you. Now, this experiment deals with specific phenotypes of corn, and we're going to follow those phenotypes through both monohybrid and dihybrid crosses. So, for example, we are using a corn cob that is a solid yellow phenotype, and this is actually an absence of pigment. And so the corn that we typically see in the grocery store and eat is a recessive type of corn that has a nice golden color, and so we usually associate corn with the nice golden color, but that's actually not the wild type of corn. This is a bred type of corn, a bred phenotype that looks more appealing and it looks corn colored, and that's what people prefer to see in the grocery store. But in fact, this is actually the wild type color of corn, this nice dark red pigmented corn. And so the pigment is in a structure in the individual kernels called the allurone, and you'll see that in the text of your lab handout. So in this cob, we have a red allurone or purple or whatever color you think of when you see that. I call it pigmented because it has pigment. And then we have a non-pigmented variety, which is what we would typically call yellow, but that's just because there's no red to cover up the yellow. So we're going to be working with these two as the parentals. So we have our nice parental pigmented versus non-pigmented type. And if you actually follow that cross in a monohybrid cross, and this is a little bit hard to see because of the plastic, so I'm going to take the plastic off. We have our cross right here. So we have our two parentals, we have our dark pigmented parental, we have our non-pigmented parental, and when we cross those two parentals, just like in any monohybrid cross, we wind up with a hybrid generation, and that hybrid generation will look all like 100% like one of the parents, and that's what we come up with as the dominant phenotype. And so that's where we see our dominant phenotype here is the pigmented. And then we take the pigmented hybrids, we cross those, and we get our F2 generation, which is down here. And hopefully you can see that it's a nice mix of both pigmented and non-pigmented kernels. And maybe if you counted them, you would actually see that it's closer to a 3-to-1 ratio because this is a monohybrid cross. Now, in a monohybrid cross, typically we are looking at the F2 generation and kind of interpreting our way back. And so what we're doing in this lab is we're taking the results of that F2 generation at the bottom, and we are going to count the kernels and see if they do in fact follow that 3-to-1 ratio. So an example of an F2 generation would be this cob. And this cob you can kind of see, like the one in the box, has a lot of red kernels or pigmented kernels and some of the yellow kernels here and there, the non-pigmented type. So we're actually going to count the offspring. Now this cob represents hundreds of offspring. That's a lot to count. And so we're actually going to take a sample from a cornfield. And so from the entire cornfield where this cross was accomplished, we're going to take a small sample that I have already pre-made. And so this sample should reflect pretty much what's going on here. Now, an alternative of what could be happening is something called a test cross. And a test cross is where you have the same parents, but in the F1 generation, instead of a self-cross where you cross a hybrid to a hybrid, you cross a hybrid to a parental, and specifically the recessive parental. And once you get at the bottom down here would be a different proportion of offspring. And those offspring are going to be more equally distributed because you had one parent that was a recessive. And so that cob would look more like this cob, although this is a really old cob. You can see that it has a more equal distribution of yellow and red in comparison to the monohybrid cross with a 3-to-1 ratio. So this is what we're actually testing for when we are counting our offspring. So in the very first experiment for the monohybrid cross, what you would do is you would take your sample, and I have a sample for all of the groups. Obviously, you're not going to be able to count these yourself. I'll make some room here. But the procedure would be just basically taking the kernels, and you can't see that in that video. It's kind of off screen. But you would take those kernels, and what you would do is you would sort them according to whether they are pigmented or non-pigmented. And you can use the parents as a guide to help you figure out and count your individual kernels. Now, I'm not going to sit here and make you watch me count all these kernels. So I'm going to count them for you, and then we'll come back to the next segment, which is the dihybrid cross. All right. Whoops. Sorry, guys. I forgot to turn off the autoplay. All right. So, haha, that was amazing. So I do have a YouTube channel that's new to me. And that is on the YouTube channel if you want to watch it later. But now you know a lot about corn. And so you should be able to do the first part of the lab dealing with the monohybrid corn now that you know how to do it. So if you go to your assignment, And blah, blah, blah. So if we go to the assignment, I'm going to share that with you real quick to just kind of go over it with you. Preview, that's what I want. So if you look at the actual assignment, you can see the directions are just there. And then, okay. So what I wanted to show you is that I counted, oops, that's not it. I counted the offspring, and they are right here in the question text. So I didn't actually like do it all for you. So you have to figure out where they go and how to fill in your table. But just like the coins, you have the probability section, and then you have the expected number that you will need to fill in for that. And pretty much, I guess you know what to do for that. So go ahead and work on it. And then when you are ready, someone can share the Dihybrid Cross video. Now that I know that the sound actually works, you just have to remember to click the share sound button all the way in the corner if that's something that you are going to share. All right. So I am going to turn it over to the class. It looks like everyone's here this morning, and I applaud you for that. Yay for making the effort to show up despite having issues with your power and Wi-Fi. I know here in Chesterfield, they decided to go back to school, but so many kids are without power that a lot of kids just aren't going to school because they can't shower. So it's just a bizarre time right now. So anyway, I'm going to turn it over to one of you, whoever wants to share. Is anybody on a computer that can share? Anyone? Okay. So someone can share. It looks like Coral says she's on a computer. She can share her application, the assignment, and then you guys can work on it together. And then once you get ready for Dihybrid, you can either let me know and I'll start the video, or someone else can start the video through the sharing function. All right, everyone good with that? You know what to do? And I'm going to be just sitting here. Yeah?
Sorry, my dog is barking.
Yeah, I was wondering if your dog was going to participate. We might have trouble understanding it.
Okay. Share content. I think, can you see this? Can you see my screen, everyone?
Not, yes.
Oh. Okay. So record whether the trait is dominant or recessive. So the numbers are what Dr. Shows have counted for us. So green is, there's more of the green and there's less of the albino. So if so this is the F2 generation. Yeah. So this
Make
should be
sure,
green
make sure
dominant
you're,
and albino recessive, right?
yeah, make sure that everyone's helping you, Coral.
Okay. All right. I'm just gonna say green is dominant because there's more.
I agree. And albino would be recessive. I agree.
Okay, good. We are green.
And number 2
Yeah,
is pretty
and when you
simple.
do, how you decide which was dominant, which was recessive above. How would y'all say that?
Basically,
Just
there's more individuals that had there's more leaves that had the green trait than the albino, so we would assume that the green was the dominant because of that.
I said something similar to Kayleen that, like, based off of the F2 generation, there was more green present than albino, so we can assume that that was a dominant one.
generation. If they have albino leaves, does that mean they'll die? Doctor shows.
That is a possibility, because that would mean that they don't have chloroplasts. But this experiment is just the shoots of the plant, so it's just to look for the phenotype. But yeah, they would not survive.
Okay. Just curious. Okay, question 3. In the following table, fill in calculated probability and expected numbers of offspring according to the data given. Raw data, pigmented 142, non-pigmented 50. So probability, observed number, expected number. Okay. So raw data would be observed number, right?
Yeah, it would be.
Okay. Pigmented 142, non-pigmented 50. Total is 142 plus 50 is 192. Okay. So F2 generation for monohybrid, that would be, probability is 3 to 1, right? For dominant versus recessive, the phenotype.
For monohybrid, yeah.
Okay, so that's 3 out of 4, and then 1 out of 4. I wanna press the screen. I don't know. I don't understand.
From our perspective, it's like, oh, never mind, you fixed it. Never mind. I was about to say, like, one of those retro
I
window
did?
things, when like it go out and it'd be like window out of window forever and infinity. That's how it was.
Mm.
We good this morning, though.
Oh.
Yeah, we good.
Okay, it's good now?
Yes.
Okay. Just wanted to.
Oh,
Okay, good. Okay.
We good.
Yeah, I was switching back to see your comments.
Oh no, yeah, I understand.
Or,
The same thing for me. I understand.
okay. Maybe I should switch it to. Hold on. Share.
If you share the Chrome tab instead of share screen, then you can go back and forth
Oh.
between the tabs.
Cool. Okay, so now you still see the yeah. Now you still see the thing. Okay, good, good, good. Awesome. All right. Cool. So expected is probability times total number, right? If I remember correctly.
So that means it, because if you multiply it, you get 106.5. And then the expected for the non-pigmented would be 12.5.
I got something different.
Nice.
I don't know if I'm doing it wrong though.
I might be, because I just,
Wait. The total needs to be still 192, right? So that was, oh, probability times 192. So what
oh,
did you,
I did about 142. So it would be 144 instead.
okay. Ah,
And then the second one would be 48.
cool. All right. So observed minus expected, this would be -2 and +2. Cool. All right. Is everyone good with the table?
Yeah.
All right. Awesome. Kernels were already counted before you could calculate the expected number of F2 offspring in the table above. Why was it necessary to complete the count total before you could enter the expected number? Count total. Oh yeah. It would be because we have to multiply the probability by the total number to get the expected.
Yeah,
Right?
basically,
Yeah,
I
that's
was
what we
thinking
did.
something along to that effect. So yeah, that's what I was thinking, something to that effect. So
Okay. Probability. I still spelled it wrong. Oh, I did. I don't know. Probability times total number. What did I do wrong? Probability expected number. The total number necessary. Necessary. Sorry, my spelling is really bad. To get the expected number because it is in the equation. if we did not know the total number, it would be impossible for us to find the expected. Okay, how's that?
I think that is fine. So Brown, what I said, mine was a little bit shorter, but I definitely think that that is fine. So great.
All right, amazing. All right. Okay, what does the deviation represent in this experiment? Deviation is observed minus expected. Observed minus expected. What would you say this represents? Like, is it like how close?
Keep going. What was you about to say?
How close it is to the expected? Like,
I was gonna say that the deviation in wait, I don't know if I'm correct, but I was basically gonna say the deviation in this experiment represents the difference between the observed and the expected. Because you subtracted. That's why I just
okay, yeah.
the difference.
I kind of see what you were saying to the core about how it represents like how close we were to the expected.
I agree too. It's a measure. Oh yeah, either one. I think either way is right. Mhm.
Kernel numbers. Okay. Everyone ready for the next question?
Yes, we can move on.
All right.
If you will, I can
Awesome.
chat for you so that way you don't have to keep clicking back and forth. Because I know like when you're on one, it's kind of hard to like kind of like keep going back and forth to check it. So I can read it out to you if you want, if it'll speed it up for you.
Okay. Thank you, Jura.
You're welcome.
Question 6. What is a procedural error in your own words? Give an example of a procedural error that may have occurred in this experiment and explain how it could change the number of offspring. Okay. What do y'all think what procedural error is in your own words?
Procedural error is like something that occurs while you're doing the experiment that may have an effect on your outcome.
Okay. Not a mistake, but error.
All right. Rhonda says, yes, there are different ways to get the same answer. Also, would be a way to make a procedural error. Bloop. Chat. Red.
Okay. Proceed. Procedural.
You're so fine. Rhonda says, I gotta step away again at work. Come on, girl. It's okay. You're making them coins. We hear that.
It's
You're making those
you're fine.
coins. Don't get weak off me. Ooh, trying to make it it's a good mood type of mood. We're good. Yeah, you're perfectly fine though. We understand. The money ain't gonna make itself. We can't let it make us.
Okay. Different ways to get to the same number. Okay. When there would be multiple ways to get the same result, but I don't know if I'm wording this okay or not.
I don't think your wording is off necessarily. I don't know if anybody agrees with me because I said I don't see anything like overall what you're saying. I think one suggestion I can make is possibly instead of making an example, make it more so like you're explaining how it could change it rather than an example. But but like I said, what you're saying is fine. Does anybody else agree with me, or does everybody else just think it's fine? Because if so, I'm gonna close my big old mouth and we're just gonna leave it be.
Because I know the question asked for an example, but I didn't know if it was like wanting one from what's this that may have occurred. Yeah, in this experiment. So
Right.
I guess one that could have occurred
Oh, and
would be
this.
like. Mm-hmm.
Okay, so it's two parts. My bad. Never mind. You're fine. I don't know if it's from the experience, but like she said, my bad. The big mouth do
One
not
that
read all that reading comprehension skills.
one that could have occurred would be like if somebody miscounted because we are counting it by hand.
Right.
in this experiment is counting errors. Miscount up a different number. How about that?
Yeah, and then, well, yeah, it doesn't ask for like a way to fix it, so that should be fine.
Okay. 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 Alright. What is a random sampling error in your own words? Give an example of a random sampling error that could have occurred in this experiment and explain how it could affect the outcome of the experiment. A random sampling error.
Yeah, kind of going off. Well, Mayana said in the chat where each sample in the experiment has a chance of being chosen, basically like a generator giving a random number. I worded this bad in the chat, like a little smiley face with the tears coming out. And then Kayleen said, taking the data from a sample, which means it doesn't reflect the population properly.
Oh. So like you're taking samples randomly in a population, but it ends up not reflecting like the ratio properly in the population.
Like something random happens within the sample group that doesn't reflect the population's data.
Did you
Yeah,
read
I like Kayleen.
Oh, never mind. You beat me to it. I was about to read it off.
Of the
I guess an example could be us not getting enough seeds to like fully see how the population is.
of random sampling in this experiment if that there was not enough average? No. Mm. Seed collected from the cornfield doesn't truly represent the ratio of the pigment. What do you think? It's okay.
I think you're fine. I'm not just being a yes man either. I'm reading what you're saying.
Cool. Say again? I didn't catch that.
Oh, I was just saying that I wasn't being a yes man either. Like I'm actually reading what you're saying because I know it's like a lot of yes, 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 yes. Like I'm not just Oprah. Like, I'm reading it. It's okay.
Okay. Oh, so we are at the end of the Monohybrid Cross. So now we need to watch the second video. Is that right?
Yes, that's correct.
Yes. Do you want me to do it, or can you? Do you think you can do it, or?
Can you do it? Because
Yep.
yeah, can you do it? All right. Let me stop sharing. Oh, stop. There. Cool.
Now, in the second part of the experiment, we're going to be working with a dihybrid cross. Now, a dihybrid cross is a little bit different from the monohybrid because we're actually looking at two different traits at the same time. Now, we're familiar with our red and yellow traits, and our red and yellow traits are obviously the pigmented allurone and the non-pigmented allurone. But now we're going to add in a second phenotype, and that second phenotype has to do with the shape of the kernels. Now, I know you're looking at these two cobs and you're saying, well, the red one's really, really tiny, but that's not necessarily the trait we're looking at. We're actually looking at the individual kernels, and we're going to try to figure out the ratio of offspring that share parental traits and non-parental traits. All right, so one of the examples I have here is a variant that's a little bit hard to see. Hold it up. These are wrinkly, and these are actually the endosperm of the kernel is sugary. And so in a normal seed corn, or the corn that you would feed to cattle or to other livestock, you might buy it for squirrels in your yard. I don't know if you like squirrels or not. They're kind of annoying to me, so I don't feed them. But this is seed corn, which is very starchy. And the reason that it stays nice and plump like this is because it's full of starch that doesn't dehydrate as much as sugar does.
And so when you buy corn in the grocery store, if you dried it out, it would actually look like this, because this is sweet corn. So the difference between seed corn and sweet corn, I know it's hard to hear the difference, but the sweet corn is sugary, and therefore when you dry it out, you have all of this, the water loss that was attracted to the sugar goes away, and it just leaves these wrinkly kernels behind. Now the other thing here that we see, so this is a yellow starchy type of corn, that's what seed corn is, and this is a yellow wrinkly type of corn, which is what the type of corn, the sweet corn that we eat is. But in addition to these two phenotypes, we also have the yellow versus red. So we have the yellow versus red phenotype differences, these two variants, and then we have the smooth versus wrinkled variants. Now the one cob, the one parental cob that, or the, I guess the F2 cob that I don't actually have is the red and wrinkly, and that's because red corn is usually not grown as sweet corn. Red corn is usually grown as popcorn, and so you can actually buy a bag of popcorn that comes from these red kernels. You can tell it's popcorn because they're tiny kernels, and they're harder than the seed corn or the sweet corn, and so it's a little bit harder to find red wrinkly corn because people won't eat it. So you have to grow it yourself, which I'm trying to do, but you know, corn takes a long time to grow. All right. And that's how I know that this is popcorn and not regular eating corn. Now, if we do the same type of dihybrid cross, like our monohybrid cross, where we have our two parents, and our parents will actually be these two. So we're looking at double traits this time. So we have pigmented and smooth, and we have non-pigmented and wrinkled. So those are our two traits. If we cross these two parents and get to the F1 generation, all of the F1 generation is going to look like the two dominant traits, which is this. So this is actually double dominant. The starchy is dominant to wrinkled, and the, smooth is dominant to wrinkled, starchy is dominant to sweet, and then the pigmented is dominant to non-pigmented, as we already saw in our other example. So that means that when we get our F2 generation, we're going to have not just two phenotypes, right? Because we're not just looking at red versus yellow this time. We're actually looking at red smooth, yellow wrinkled, red wrinkled, and yellow smooth, right? So there are 4 different phenotype combinations that we're actually looking for. And if you were able to see this cob really well, you would see some of each. So you can kind of tell the difference between the yellow smooth and the yellow wrinkled. The red wrinkled are harder to see, but you can see a lot of red here, and some of those are red wrinkled as well. So I always say that the wrinkly ones look like teeth, like molar teeth, you know, in the back teeth that are really wrinkly. So for this experiment, we're going to be counting the F2 generation. This cob represents an F2 generation, but again, it's hundreds of offspring. Obviously, the more you actually count, the closer you are going to get to Mendel's ratios. But in this case, again, I've set up some samples of our field of corn for you. And if you were in class, what you would be doing is you would take your sample, you would open it up and dump it out, and then you would start sorting it according to the different double phenotypes. And so you would have some, let's see, red wrinkly, you would have some yellow smooth, you would have some yellow wrinkly, and then you would have purple smooth. And it's funny that that was the harder one to find, because that's the one that should be most common because it's both dominant traits. And so if you remember your dihybrid cross Punnett square, then you're going to remember what that ratio is supposed to be. It's supposed to be 9, 3, 3, 1, where you have 9 and 1. 9 would be the double dominant, 1 would be the double recessive, and then you'd have 3 of the recombinants of one type and 3 of the recombinants of the other type. So I'm not going to make you watch me count these. I'm going to count them, and I'm going to present the data for you in the lab as we talk about the lab. All right. So this is, hopefully you've got an idea of the procedure so that you'll be able to answer the questions and do the analysis.
Ha, I got it before the next video played. All right, so that is the dihybrid cross, and so anyone can go ahead and start sharing to finish up the lab. And again, the results should be in the question text for the dihybrid. Questions? Cool.
Want me to share it again?
That's up to all of you. I mean, you know how to do it, so it'd be easier, but I don't know who's on computers, so.
Rhonda says, yes, if you could. Yana says, please. Please, with the crying emoji. And Rhonda says, I have a narrator. Period.
All right. Awesome. All right. Chrome editing to share. Yeah. Cool. Okay. In the following table, indicate which phenotype combination parental and which are non-parental in the F2 offspring. So our two parentals are the double dominant and the double recessive, right? So that would be pigmented and smooth. Cool. Okay. Dominant is the smooth and pigmented. So pigmented and smooth is parental. And the other parent is double recessive, so it would be wrinkled and non-pigmented.
Cora, I'm not sure if you want us to, but if you don't, you want to keep it still, that's fine too. But we can't see your screen. Never mind.
Oh.
Bloop, it's
How about
here.
now?
Never mind. I can see it now.
Okay, okay.
Good. All right.
Good. All right. All right. How about now?
Oh, yes, we're good.
Okay, good.
And Faith
All
says,
right.
I'm
So
driving
we got our
right now. Faith, you better not be sitting here looking at this thing and driving, girl. You better focus on that road.
yeah. Stay
Like,
safe.
with it.
Distracted driving.
Only you can prevent distracted driving. We're not giving it up like that. I want you to make it to your destination safely.
Yes. All right. So now we got the two parents. That means the other ones, the two leftovers are non-parental, right? No. Non-parental. Non-parental. Pigmented as a dominant and recessive, and as recessive and dominant. All right. Everyone got that?
Yes, we're good.
Oh,
Oh, period.
amazing.
Rhonda says, I'm doing a Punnett square now for that one. Period. I love it.
Amazing. Allurone color trait, we will use gene symbol P. So the big P allele encodes pigmented, and the little p allele encodes non-pigmented. For the starchiness trait, we will use gene symbol S. So the big S allele encodes smooth, and the little s allele encodes wrinkled. Select all the genotypes that will lead to the phenotype pigmented and smooth. Okay. Y'all ready for this? Pigmented, which is dominant. So any one of them that has big P in it will, any one of them that has big P and a big P, at least a big P, and at least a big S, right?
Okay.
Yeah.
Miss, I like your last name a lot, so I'm gonna say your last name. I don't know. I'm sorry, everybody's getting a special way I say their name. Miss Teasley is saying big P, big S, and big P, big S, little S. Is it okay if I say Miss T like that?
Big and little. Big, little, big, little. No, big, little, big, little. Okay. That's it. That's it. Yeah. Okay. Smooth as big as. Okay. That right?
Rhonda says, I believe so.
Amazing. All right. Next one. Sorry, I don't know how to talk to people anymore.
Girl, no, sharing your screen and Blackboard Collaborate do something to you because I know I was talking all over the place when I was sharing mine. You're so fun. I don't know what it is.
Okay, select all the genotypes that will lead to the phenotype pigmented and wrinkled. Pigmented is big P, so any one of them that has at least one big P, and then wrinkled is recessive, so at least one big P and two little S's. So
Teasley says big P, little S, little S.
all right.
Dr. Shows says, just letting you experience my role for a little while. So, and Miss Teasley said, for real, Miss Teasley says big P, little S, and Miss Drakeford. Oh, Miss Drakeford says big P, little S as well. And Rhonda says, LOL, the struggle is real. It really is. It is tough. It is tough.
All right, So we got, I think that's all, right? Yeah. Yep. 2 of them. All right. Cool. Next. All of the genotypes that will lead to the phenotype, non-pigmented, so that is little p, and then smooth is at least one big S.
Miss Teasley says little p, big S, little s, and then little p, big S, and Miss Strakeford agreed. I feel like what I'm saying,
Cool.
I cannot say that. That is not appropriate for class. Never mind. It was a TV show.
Oh,
Never
not this one.
mind.
Wait. Sorry. Little p, big S, big S. Yeah. Little p, big S, big S. All right. Yes. Okay. To go back to the chat to make sure. Yep. Okay. Okay, we're good. Next, all the genotype that will lead to the phenotype, non-pigmented, so little p, and wrinkled, little s. Yeah. Just the one. Double recessive. It's the 1 in the 16, right? Cool. That's it. That's it. Okay. We good?
I am believing so. And Rhonda says yes, thank you. She said the abbreviation, but I just spelled it out for you that way. Because I'm so tech savvy.
All right. Cool.
Rhonda says LOL.
Oh, no.
Oh my God. And LOL stands for laugh out loud if you didn't know. Girl, they should choose me for the next series. People would get their laughs.
So I already
All right.
did the table. I think I did it right.
Oh, awesome.
So for the probabilities, wouldn't it be the 9-3-3-1 rule?
I think so.
Yeah, so it'll be 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16. And then for the observed, oh, sorry. Well, yeah, it's on the top of it.
Ten.
It's
The total number
278. It's 278. And then the expected for the first one is like 156.4. 52.1, and then 52.1 again, and 17.1. And that works out to like 277.7, so like about 278.
All right. Okay. Deviation. Deviation. All right. So that would be observed minus expected.
I got 7.9 for the next one. The one, the pigmented slash wrinkled.
Okay.
52 minus 52.1 minus, let's say, yeah.
Okay. And then,
And Rhonda says minus 7.1 for the next one.
all right. Yeah. About to zero. They weren't pretty numbers. So everyone got that? Got those numbers.
Mm-hmm.
I'll just keep it at the top.
Yeah, we're good. Miss Teasley said yes. And Kayleen says pigmented smooth were dominant.
Okay. Pigmented. How your own and smooth for endosperm starchiness were dominant. Two phenotypes are dominant, blah, blah, blah. Alright. Yep. Okay. Alright. Got it. Recessive.
Kayleen says non-pigmented wrinkled for recessive. I think she said respective. I think she meant to say recessive. I think. Correct me if I'm wrong.
How you run and wrinkled
Kayleen says yeah.
endo,
Laugh out loud.
endosperm starchiness were recessive. All right. All right. How did you determine the probabilities for the different phenotype combinations that you entered into the dihybrid cross table above? How did we determine the probabilities? Dihybrid.
Kayleen says based on Mendelian genetics, and Rhonda says the 9331. Kayleen says I have more to say. Bad. Rhonda says and dihybrid cross. Kayleen says my mic won't work.
Kayleen.
All good.
Okay. Determine the probabilities for the different phenotype combinations based on the Mendelian genetics. Mendelian genetics. Dihybrid cross. How do you spell that? Mendelian genetics. Dihybrid cross. Which has a ratio of 3, 1. How is that? How's that?
I personally think those are fine. And Rhonda says perfect. Thank you. And also, before we submit, can we go from like 9 back to 9 through 12? So I can just make sure that mine was.
Yeah.
And I was kind of like reading the chat sub too, so I know like it was kind of a little bit off. Let me see. I'll take pictures of that.
Yeah.
I don't have to hold everybody up. Okay, you can go down.
All right. 10.
Okay, you can keep going down. All right, and you can keep going down. All right, thank you. I'm about to go fill that in right now. That was the only thing I really can.
All right. Okay. Anyone else want to look at specific
Rhonda says, "Thank you so much, Coral." Period.
You're welcome, Rhonda. My pleasure. All right. So everyone has all their data and numbers and answers they have. What time is it? 10:19. We're doing good. Okay. If everyone is good with the answers, I will stop sharing. Stop sharing. Stop. Yeah. Thank you so much for your cooperation.
Oh, look at that. You guys finished early, huh? Good job.
Thank you.
Have a great day, everyone.
Have a great day.